Welcome to Botany class. Previously, you learned the process of linkage. Let's move on today's learning objectives. Linkage groups, crossing over, importance of crossing over, recombination, recombination frequency, genetic mapping, map distance and its uses, multiple alleles. These are the topics we are going to cover today. Let's start with linkage groups. Linkage groups. What's meant by linkage groups? Groups of linearly arranged linked genes are called as linkage groups. You know that when you learned about linkage, you will know these terms. What are linked genes and so on. So, linkage groups. Today's topic is linkage groups. Definition is groups of linked genes, linearly arranged linked genes are called as linkage groups. Where the genes are located? The genes are located within the chromosome. So, within the chromosome there are genes. What are linked genes? The genes which are arranged very closely is inherited together. So, these genes have a possibility to link it, inherit together. So, linkage groups are known as groups of linearly arranged linked genes on a chromosome. Groups of linearly arranged linked genes on a chromosome is called as linkage groups. The linkage groups in a species, in an organism, the linkage groups corresponds to the number of haploid sets of chromosome. There are some examples. These are the organisms: Mucor, Drosophila, sweet pea, Neurospora and maize. In these organisms, the linkage groups, number of linkage groups, these numbers corresponds to the haploid set of its chromosome. In Mucor, the linkage groups are 2. So, the number of haploid set of chromosome also 2. Likewise, Drosophila is 4. The number of linkage groups are 4. So, the haploid set of chromosome also 4. And sweet pea, the number of linkage groups are 7. And the haploid set of chromosome also 7. Neurospora and sweet pea, it is 7. And maize, it is 10 in number. So, all organisms, in these organisms, the linkage groups 2, 4, 7 and 10, which corresponds to the number of its haploid set of chromosome. Today, you are concentrate the main topic as crossing over. So, before that, this linkage and crossing over, these two are different process. Linkage and crossing over is a different process. What's the difference? There are certain differences seen in linkage and crossing over. Linkage, you already learned in the previous class. The definition of linkage is very closely arranged genes like this in a homologous chromosome. When the genes are very closely arranged, it tend to inherit together. Such genes are called as linked genes. So, there is a possibility of closely related genes which inherit together. There is no separation. It inherit together. But in crossing over, it separates. See the differences. What are the difference between linkage and crossing over? Number 1, the genes present on chromosomes stay close together. In linkage, the genes, this is gene 1 and this is gene 2. Gene 1 and gene 2. So, in linkage, the genes present in chromosome, the present on chromosome stay close together. But what happens in crossing over? Crossing over leads to separation of linked genes. These two linked genes are separated when crossing over taking place. That's the first difference. What are the difference? Linkage, the genes present on chromosome stay close together, but in crossing over it leads separation of the genes, linked genes, separation of the linked genes. Second point, linkage involves same homologous chromosome. Within the same homologous chromosome, the genes are linked. It's a possibility. But in crossing over, there is a exchange of segments. Exchange of segments between non-sister chromatids of a homologous chromosome. This is one chromosome. This is another chromosome. So, these two, this chromosome, this is a homologous chromosome, this is another. These chromatids are known as sister chromatids. But these two chromatids are called as non-sister chromatids. Sister chromatids, non-sister chromatids. What's the difference? Exchange of segments between non-sister chromatids of homologous chromosome. This is one homologous chromosome, this is another homologous chromosome. In crossing over, exchange of segments, this segment and this segment, which exchange its character in crossing over. That's the second point, second difference. Third difference, in linkage, it reduces the new combinations because the non-sister chromatids which do not separate, exchange its characters. So, here there is no new combination which is related to the parental one, or these genes are inherited together as it is in the pair of chromosome, homologous pair. But in crossing over, it increases new combination, increases the variability by forming a new organism which increases. So many variations are taking place because the non-sister chromatids are exchange its chromosomes. That's the difference between linkage and crossing over. There are 3 points very important for 3-mark question. First point, genes present on chromosome stay together in linked genes, but in crossing over it separates. What's the second point? It involves, linkage involves same homologous chromosome, but crossing over the segments, the exchange of segments between the non-sister chromatids, and in linkage reduces the new combination, but crossing over increases the new combination. Then let's move on, crossing over. Very important 5-mark question. Crossing over. The term crossing over was introduced by or coined by Morgan in the year 1912. What is crossing over? Crossing over is the process by which exchange of genes from the non-sister chromatids. The genes are located here. Here, if it is A, B, here small a, small b, small a, small b. In crossing over is the process by which exchange of chromosomes. So, inside the chromosome there is a DNA segment, DNA. So, from this DNA sequences, there are number of genes located in a particular locus. So, this locus, in this locus, the genes here and here, which are exchanged from the non-sister chromatids. So, it increases the new recombinant genes. It produces new varieties. That is the process of this. When it takes place, when crossing over takes place, at the time of Pachytene stage of meiosis. Before study this mechanism of crossing over, let's see what are the stages in meiosis, because crossing over occurs at the Pachytene stage of meiosis. Meiosis, which takes place in two stages, meiosis 1 and meiosis 2. Let's write like this: meiosis 1 and meiosis 2. So, this crossing over takes place, mostly it takes place in the germinal cells. Germinal cells which produces the gametes. So, it occurs in the germinal cells. Rarely it produces it takes place in the mitotic cell division. So, this meiotic division, Pachytene stage is the stage of meiosis. Meiosis in the cell division, let us recall what you learned in the 11th standard. Meiosis 1 and meiosis 2. Meiosis 1, which consists of prophase 1, metaphase 1, anaphase 1 and telophase 1. And this meiosis 2, which also contains prophase, metaphase, anaphase, telophase 2. Here 2. Everything it comes the same stages. But in this prophase 1 again classified into 5 different stages. They are leptotene, prophase 1, leptotene, zygotene, pachytene, diplotene and diakinesis. So, this crossing over occurs or taking place in this stage, Pachytene stage of meiosis 1. So, this stage comes under meiotic 1 of prophase 1. From prophase 1, among these stages, this Pachytenes occur. So, during this stage, crossing over, the process of crossing over taking place. Let us move on the mechanism of crossing over. Mechanism of crossing over. Mechanism. When you see the mechanism of crossing over is precise mechanism. So, it comes under 4 different stages. So, crossing over starts from synapses. What are the stages involved in this? It involves synapses. Number 1, the first process, synapsis or pairing. Synapsis or pairing. Second process is Tetrad formation, and third stage, first synapsis, it involves the stages crossing over. Topic is crossing over, mechanism of crossing over. Synapsis, Tetrad formation, crossover and terminalization. These are the 4 stages involved in this crossing over, mechanism of crossing over. Let's see one by one. First, synapses. What's meant by synapses? At the time of cell division, this meiotic cell division, first in this stage, prophase stage of zygotine, in this zygotine stage, this synapse is taking place. What is synapses? It's the intimate pairing of chromosomes, the homologous chromosomes which pairs. During this stage, there are different, you know that in the nucleus, number of changes takes place. In this particular stage, zygotine, what's the change? The homologous chromosomes starts to pair. It comes closer. It aligns side by side. The homologous chromosomes align side by side and it produces bivalence. So, at the time of synapses, it produces, now this chromatids are called as bivalence. Bivalence. Two chromatids, the homologous chromosomes are pairing. Pairing begins in this stage, zygotine stage, at the time of meiotic process, prophase 1 of zygotine. It initiate, during this stage, initiation of pairing. Pairing is called synapses. So, the homologous chromosomes are pairs. It comes, it align closer. The homologous chromosomes align together very closely. And now this chromatids are called as bivalence. So, bivalent formation occurs during this stage, synapses. Then what's the next stage? The tetrad formation. After producing this bivalent, now this chromatids produces a tetrad. Tetrad which consists of 4 different chromatids. So, this is a homologous chromosome. This is one homologous chromosome. This is another homologous chromosome. Homologous chromosomes. So, this bivalence now becomes tetrad. The 4 different chromatids are formed. So, this is tetrad. Now, this stage is called 4 chromatids. Each homologous chromosome which contains 4 chromatids. These are the identical structure or similar structures of chromosome. This is called chromatid. So, this chromatid joined at a particular point called centromere. Centromere. All these chromatids are joined at a particular point called as centromere. Now, this chromosome is known as tetrad. Tetrad formation. When it forms the tetrad at the time of Pachytene stage. So, crossing over takes place at the time of Pachytene stage. In the bivalents are formed at the time of zygotine stage. All these stages are in the prophase 1 of meiosis. So, I hope that you remember what you learned in the meiotic division of previous class, that is 11th standard. Just recall what are the process takes place in zygotine and Pachytene. So, bivalence, each word is definition. What is bivalent, what is tetrad formation and what is synapses. So, synapses is pairing of genes. Pairing of chromosomes, pairing, synapses. Tetrad formation, 4 different chromatids are formed which is connected or joined by a particular point called centromere. So, this is the centromere, 4 different chromatids are formed. For example, in this there are the chromosomes which contains gene, capital A, B, here small a, small b, small a, small b. So, each chromosome which contains the genes. At first, before this chromatid, what happens? Synapsis in the chromosome, it pairing of chromosome, that is synapsis. Then tetrad formation, the 4 different chromatids are connected in a particular point, centromere. Now the genes located in a particular locus, this is one locus, this is another locus, particular locus, the genes are arranged linearly in a chromosome. So this tetrad is formed. Next process is crossing over. What is crossing over? Now these chromosomes which contains, this is sister chromatid, these two are non-sister chromatids. When this non-sister chromatids, these two non-sister chromatids exchange its segment. How it exchange? Look at this diagram. Here, this is a chromosome, tetrad, and this segment, these two segments connect at a particular point. This segment, these two segments, non-sister chromatids, this crossing over takes place at the stage of Pachytene stage of prophase 1 of meiosis. The non-sister chromate, here the chromosome is, here the gene is A, B, here small a, small b, small a, small b. These are the genes, the location of the genes are this. What happens during crossover? At a particular point of this non-sister chromatids, it joined. The non-sister chromatids are joined at a particular point. Now this point is called as chiasmata. Chiasmata. In this particular point, now it produces a cross-shaped or X-shaped structures where the segments are exchanged, the genes which exchange. Here there is a break at the point of chiasmata, the segments break and exchange. So breaking and rejoining of genes taking place during this stage at the time of crossover. As a result, it produces a recombinant gene. This related to the parental one, this also related to the parent. But here when crossover take place, exchange of segments. So it becomes, this forms, here it is normal, this also normal, but here it produces half from this. Here it produces this normal, here. Here it gives half this. So how it exchange its character? Here AB, here A, but this segment, this small b here and this capital B here, here is small a, here small a, small b. So now these two genes are called as recombinant gene. These two genes are named as recombinant. Now next process of mechanism of crossing over is terminalization. What's meant by terminalization? Now this chiasmata, the chiasma which moves towards the terminal end of the chromatids and the homologous chromosomes are separated. So now when the homologous chromosomes are separated, it becomes, this is a homologous chromosome and this is another homologous chromosome. So it separates AB, A, small b, small a, capital B, small a, small b. As a result of this, it produces 4 different genes. These two are recombinant gene and these two resembles as a parent. So how it shows, the first one, it gives the parental one, this. Now terminalization, these chromosomes terminalization, the chiasmata, at the region of chiasmata, the segment breaks and exchange during crossing over. At the time of terminalization, chiasma move to the terminal end of the chromosome and now it is, as a result, get different recombinant genes. This first and last segment which resemble to the parent, these two are recombinant. So this consists of capital A, capital B, and this segment consists of capital A and small b, and this one consists of small a, capital B, and this one resemble to the parent, small a, small b. Now these two are parental one and these two are recombinant gene. So this question very important, 5 mark. Mechanism of crossing over, which involves 4 different stages: synapsis, tetrad formation, crossover and terminalization. As a result of this, recombinant genes taking place. What's the importance of crossing over? In crossing over, in this process, there are exchange of segments. So these segments are exchanged, interchange of segments. Due to this, it produces a new variety. So it leads to evolution. And in the plant breeding, this new useful combinations, sometimes it produces a new. If it is new, when the characters are mixed together, it produces new varieties. So it is involved, it is very much useful in plant breeding techniques. And this crossing over studies which reveals that the genes are arranged in linear order. For studying about genes, this studies of crossing over reveals how the genes are located in the chromosome. And genetic map, which made based on the frequency of this crossing over. Based on the frequency of crossing over, genetic maps also made. So these are the importance of crossing over. Importance, differences, these all questions are very important for exam oriented. Then what is meant by recombinant frequency? The recombinant frequency, which is represented as RF, recombinant frequency. What's the definition? Recombinant frequency. Recombinant frequency or RF is the percentage of recombinant genes in the organism. In an organism, the percentage of recombinant progeny is called RF. So it is calculated by a formula. RF is equal to number of recombinants number of recombinants divided by number of offsprings. Number of offsprings into 100. So, what is recombinant frequency? It can be calculated by this formula. Number of recombinants, this number of how many recombinants are formed divided by number of offsprings. Out of this 4, how many recombinants are formed? So, here out of this 4 chromatids, 2 are recombinant, total number is 4 into 100. So, the percentage RF recombinant frequency. Then, genetic mapping. What's meant by genetic mapping? You know very well, the genes are located within the chromosome in linear order, and each chromosome have a specific locus. This is one locus, this is another locus, or position, which is named as loci. Locus. In the locus, here it's the genes are located within the locus. Gene mapping or genetic mapping is the diagrammatic representation of the arrangement of genes and the distance between the genes. That is genetic mapping. And what is meant by map distance? The unit of distance in a genetic map is called map unit. So, this map unit can be calculated or it is measured by MU. Map distance, which is calculated by 1 map unit is equivalent to 1% of crossing over. So, also it is called as this MU, or it can be measured by centimorgan. To honor Morgan, the unit is called as centimorgan. For example, if the distance, map distance to be, there are in a gene, if you are here there is a gene which contains A, B, C and D. So, it can, here there is a gene here and this gene and this gene. So, if it is 3.5, the distance is 3.5, it can be measured by 3.5 centimorgan, or it is map units 3.5 units, or it is 3.5 centimorgan. Likewise, it can be, the map distance can be calculated, measured. Then, multiple alleles. What's meant by multiple? What's the use of, before that, what's the use of this genetic mapping? This genetic mapping is used for identify the location of the chromosomes. It is a genetic mapping definition is diagrammatic representation, which shows wherever the genes are located. For gene study, this genetic mapping is very much useful. And overall complexity of particular organism, overall complexity of the characteristics can be studied by this genetic mapping. Then another question is there, multiple alleles. Normally, in a gene which contains an allele, the pair of genes are arranged. The pair of genes are called as alleles. So, here the genes are located in pairs, which is represented in alphabets AA or capital A, capital A, capital B, capital B. So, multiple alleles, usually or normally a single allele is present in a locus, but more than 3 or 4 is present in a particular locus, it is called multiple alleles. You know the multiple meaning multiple, many, more than 3 or more present in the same locus is called as multiple alleles. What are the characteristics of multiple alleles? These multiple alleles always which occurs in homologous chromosomes, and this always occupy the same locus. Multiple alleles always occur in the same locus. There is no crossing over. In the multiple alleles, there is no crossing over. And if it is wild type, wild variety which exhibit only the wild type, there is no interchange. But mutant varieties which gives the interchange, always it is mutant varieties. Either it is mutant genes, mutant varieties, it produces either mutant or the wild type. So, any mutant multiple alleles are crossed, the phenotype is always, if you are crossing between mutant alleles, in multiple alleles, mutant alleles are crossed, it produces only mutant. But wild type, it exhibits always the dominant. See, mutant alleles which maybe produce the mutant or it produces the wild type which produce only the wild type. That is the characteristic features of multiple alleles. So, let us recall what you learned today. What are the topics you covered today? Linkage groups, crossing over, importance of crossing over, recombination, recombinant frequency, genetic mapping, map distance, multiple alleles. Learn all this question, draw all the diagrams in the class note and learn daily portion without painting. Thank you.